From the humble beginnings of deciphering the iconic double helix structure of DNA in 1953 by Watson and Crick, our understanding of genetics has undergone revolutionary advancements. Central to this evolution is DNA sequencing, the process of determining the precise order of nucleotides within a DNA molecule. The journey of DNA sequencing started with the groundbreaking Sanger sequencing method in the 1970s, a technique that would later facilitate the first complete sequencing of an entire genome, that of the bacteriophage MS2 in 1976 by Professor Walter Fiers at our university, the University of Kent. But it was the Human Genome Project, launched in 1990 and completed in 2003, although it kept getting completed and it was now latest published in 2022, and there was an additional completion as late as this year in 2023 that truly highlighted the potential of DNA sequencing. As a collaborative global endeavor, the Human Genome Project provided an unprecedented map of the entire genome, comprising more than 3 billion DNA base pairs. Second-generation sequencing, also known as next-generation sequencing, represents a transformative leap from the original sequencing methods. Unlike the first-generation technique, NGS platforms can process millions to billions of DNA fragments simultaneously, drastically increasing throughput and decreasing cost per base. All NGS techniques share a fundamental principle. They sequence vast numbers of small DNA fragments in parallel. Second-generation sequencing typically involves template preparation, sequencing, and imaging followed by data analysis. Brand names that you might recognize are Solexa, 454, and Solid. It was in the middle of the NGS era that NXTGNT was founded in late 2008. NXTGNT was initially centered on two leading next-generation sequencing technologies, namely the Genome Analyzer 2 from Illumina and the Genome Sequencer 454 from Roche Life Sciences. The primary goals of NXTGNT were to establish a unique sequencing platform by the integration of sequencing methods with the existing research expertise in bioinformatics, molecular diagnostics, and translational medicine. The NXTGNT team consists of a steering committee supplemented with an operational team consisting of both junior and senior researchers. 14 years, 6 months, and 6 days later, third generation is on the horizon, also known as long-read sequencing, that is distinguished by its ability to read much longer DNA or RNA molecules than its predecessors. By providing longer reads, third-generation sequencing can bridge gaps in genomic regions that are difficult to sequence, such as repetitive regions, leading to a more complete and accurate genome assembly. Oxford Nanopore Technologies, or ONT, sequencing uses a DNA or RNA strand that is threaded through a protein nanopore embedded in an electrically resistant membrane. As nucleotides pass through the nanopore, they cause characteristic disruptions in the current, which can be translated into a DNA or RNA sequence. ONT can sequence extremely long reads, sometimes over more than 2 million base pairs, offers real-time sequencing, and is highly portable, like their Minion device. Error rates are higher compared to second-generation methods, but like PacBio, they are typically random and can be corrected with higher coverage. Within NXTGNT, we realized that competing on price leads to a race to the bottom, where business continually undercuts each other's, eroding profit margins and potentially compromising the quality of goods and services. NXTGNT, in contrast, chose to compete on value, emphasizing the unique benefits and experience our service provides to its customers' collaborators. This approach generated loyalty and encouraged sustained growth. The Expertise Center for Sequencing UGent combines infrastructure and expertise for massive parallel sequencing from the consortia of NXTGNT and CMGG. These consortia consist of the following research groups that have working closely together. Lab FBT from the Faculty of Pharmacy, of which the sequencing expertise is focused in NXTGNT, with infrastructure that entails MySeq, NextSeq 500, HiSeq, Minion, Covaris, IPStar, Tecan Freedom, and Evo 200. BioBix delivers the expertise in bioinformatics, integration of data, analysts combined with a plethora of high-throughput applications. The Center of Medical Genetics brings expertise also in bioinformatics, exome and genome sequencing, functional genomics, and epigenetics. Now let's take time to focus on the different application domains this technology can be used. The first domain that I will zoom into is genomics. In the genomics part, we focus on DNA sequencing, de novo genomic sequencing of organisms, exome sequencing in humans or other animals, design and validation of targeted panels like exomes, both in model and non-model species, targeted gene sequencing, panels focused on specific diseases, shallow genome sequencing, copy number alterations. These methods can be applied in many different, for many different questions, finding causal genes for diseases, identifying novel mutations and variations that are important for certain traits. We apply it extensively in cancer research, and more recently we also have been applying it in the area of metagenomics, where we study the genomic material from environmental samples, gut samples, soil or water, and we identify and characterize microbial communities. Transcriptomics. Transcriptomics is also known as the domain of identifying all the transcripts or RNA that are present within a cell. So they are produced under very specific circumstances, under very specific conditions, or during very specific genes. RNA comprises of messenger RNA, microRNA, long non-codings RNA, which can all be sequenced and quantified using differential gene expression analysis. Our methods also allow to identify new splice variants like alternative splices, which can then all be applied to, again, very specific translational questions like cancer transcriptomics, where we analyze to see unique expression profiles in tumors, can also be used, for instance, in virology to sequence RNA viruses, understand viral evolution, understand host-virus interactions. We also have been applying these methods to identify integration sites when viruses integrate in the host genome to see if they disrupt essential components of the host defense. A next application area where we can apply our technology is epigenomics. Epigenomics describes all the dynamic alterations that affect the transcriptional potential of a cell. The main epigenetic mechanisms are DNA methylation and histone modifications. If we look at an entire genome from a single base pair to the full potential of the 3 million bases, we can characterize, as described earlier, all these individual nucleotides with a genomic method, which can be whole genome sequencing or at the other extreme end, PCR. When we look at the flip side of this coin, all these genetic methods can be complemented with an epigenetic methods. We have whole genome bisulfite sequencing, and we can also apply it on a single base resolution level by MSP. In the middle we have panels, or the equivalent of exome sequencing. NXTGNT has all the tools to analyze these epigenetic mechanisms by an integrated analysis, and this within the ultimate resolution of a single base pair or on the whole genome level. The methods that we employ are methyl cap sequencing, MIDIP sequencing, ChIP-seq sequencing. We can do ATAC sequencing and apply this on a very complicated set of samples. The fourth application domain in which we can apply our methods are the domain of the proteins, proteomics, which includes studying the entire set of proteins in a sample, leading to insights about protein structure and function, interactions. A first method that we employ is ribosome profiling, also known as RiboSeq or ribosome sequencing. It's a method to monitor the positions of actively translating ribosomes on mRNA on a genome-wide scale. We complement this method with proximity extension assays, or PEA technology, that is a technology behind the Olink Explore platform and the Olink Target 96 and Target 48 panels. It enables high-throughput multiplex immunoassays of proteins using minimal volumes of serum and plasma, or almost any type of biological sample. Let's switch gears from the methods to the actual samples used in these methods. Single-cell analysis is a cutting-edge technique in the field of biology and medicine that aims to study individual cells in isolation, rather than analyzing bulk samples which provide an average response of many cells. Since individual cells within seemingly identical populations can show significant variations in terms of gene expression and protein levels and subsequent metabolic activity, analyzing cells on a single cell level can provide insights that are masked in conventional bulk analysis. Spatial genomics, often used interchangeably with spatial transcriptomics, refers to a set of techniques that aims to measure gene expression while preserving the original spatial context of tissue samples. In other words, it combines molecular profiling with the precise location information within tissue. This spatial resolution adds an extra layer of information that traditional sequencing methods, which involves homogenizing tissue samples, can't provide. We provide all single-cell analysis and linked read sequencing solutions offered by 10x Chromium system and the GEMCO technology. Oxford nanopore sequences unamplify DNA and RNA, eliminating the need for amplification steps and the resulting quantitative and qualitative amplification bias. It can generate long reads up to 100 kilobases with a direct detection of epigenetic changes on both RNA and DNA, providing a tremendous advantage in de novo assembly, detection of large structural variants, RNA isoforms, and so forth. We are currently involved also in writing grants to acquire an Aviti machine and a PacBio Revio system. Our Palm microbeam is designed to give you advanced laser microdissection capabilities for subsequent DNA and RNA analysis, whether from archived material or live cells. It can also be applied to cryosections and FFPE tissue, even from standard glass slides. NXTGNT is specialized in low input samples, such as single cell samples, FFPE samples, that can all be handled by our laser capture microscopy. Bioinformatics is a fusion of biology and computational science, and it plays a pivotal role in modern biological research. NXTGNT provides the essential tools and frameworks to mine the generated datasets for generating meaningful insights and enabling the discovery for novel genes, pathways, and associations. In essence, bioinformatics at NXTGNT bridges the gap between massive biological data and actionable scientific knowledge, propelling advancements in biomedicine and biotechnology. We are very experienced in applying all the methods above in pharmacogenetics, which provides molecular insights connecting physicians and their patients to the latest treatment approaches, thereby enabling precision medicine today. If you are looking for more inspiration, browse our library of papers resulting from over a decade of collaborations, or contact some of our more prolific PIs. NXTGNT is a solid, experienced collaborative partner for non-standard research questions where expertise has been combined to overcome obstacles.